Now we're going to talk about the effect of stress on memory. And some of you may already know that long-term stress or extreme stress can definitely have an effect on your recall. And in some cases, it can even make a difference in your ability to form new memories. So let us take a minute and go to our notes and start thinking about stress. When is it that it really emphasizes your memories and kind of seals them in? And when does it make it hard to form new memories? And when is it like hard to recall things, at least temporarily? And flashbulb memories are kind of memory that I want to talk about for a moment. First, have you ever experienced something in your life that was so deeply emotional and you can remember everything about that time? You can remember what things smelled like. You can remember lighting. It's like imprinted so deeply in your mind's eye that it's imprinted. And this kind of memory typically is one that has an emotional connotation to it. And we're going to talk a little bit about how that could potentially happen and in a few minutes, and we're going to be talking about things like the cortex, the hippocampus, and also remember that limbic system, other types of structures that are associated with emotion as well. So, forms of memory first. Let's just review this real quickly. And again, you know this from other courses, I am sure, and the short-term memory versus long-term, and then there's working memory. Short-term memory that you remember just long enough to complete a task, and then you try to disregard it. Remembering a phone number long enough to dial it and then dismissing it. Long-term, that gets placed in your cortex and can be retrieved later on. And while you actually are working or utilizing that memory, that is engaging your working memory. So also we have implicit versus explicit, or procedural versus declarative, same things. Implicit, one of those things that you learn and you don't even consciously think about it anymore. In fact, some of those things, if you try to think about it and explain or pull it out into pieces, actually becomes complicated for you. So a lot of the motoric things that we do are similar to that. If you learn how to do something that is like probably processed in large part by your cerebellum, that it's very hard in the end to be able to explain to somebody. I'm not a tennis player, but if somebody were going to try to teach me exactly what to do to serve, I'll never forget the very few times I ever attempted to play tennis were high school, in which we really weren't trying to play tennis. Don't know if any of you had that kind of situation, but you're more interested in figuring out ways not to do what you should be doing. Or just a couple of other times when I happened to be with somebody and we tried it. And I was so excited the first time I served and I actually did that sound without trying. I'm like, oh my gosh, not that it was a good serve, but I got an sound. So that anyway. But those kinds of things that you can't explain, you can't declare, like you can, what's your address? Declarative memory. You're using your declarative memory and you can say those particular things. What time is your next class? What class is it? Declarative, explicit. And we have the transferal of memories from short-term, just immediate, to long-term. And when we're talking about the transferal of something that you're learning consciously until it becomes implicit, that's when I'm talking about. Sometimes it's difficult to mix those two up so that you can explain something that's so implicit. So what are we talking about? I already briefly mentioned we're going to be talking about the hippocampus. Think of the hippocampus as being like the keyboard. And now I'm borrowing all of this from Sapolsky. I take no credit. But the declarative part of it, the declarative, like, let us type in what it is we want to later on retrieve. And the hippocampus is where we have consolidation, where we take the things that we're trying to process and consolidate them so that we can later on draw from them. And it's via your hippocampus that you place and then are able to access memories from your cortex, which we're going to consider to be the hard drive of your wonderful computer. So your cortex, where memories are stored, particularly the declarative form of memories. The cerebellum, as I mentioned before, is a site for many implicit memories to be formed and stored and to become automatically programmed. But there's other diffuse sites as well. Too many for us to talk about right now. Just pay attention to these particular ones that we just talked about. And the reflexive motoric actions that we see so often in the cerebellum can persist despite the inability to have memories that you're even conscious of, that you're not aware that you even remember how to do it. The case of HM, brilliant case. I mean, this was such an important milestone in understanding the brain and neuropsychology. I'm pretty sure that you have talked about HM in intro psych. Certainly in a couple of my classes, neuropsychology, fundamentals of human neuropsychology, a little bit more in detail. But he showed very clearly how you can remember certain things and not even be aware of it. But then in terms of forming new memories, that becomes impossible. In the case of HM, it would be like after he had his brain surgery to try to correct or at least control his seizures. I'm going to just briefly give you like an overview in case you've never heard of HM. But HM had irretractable seizures. They were getting worse and worse and worse. Medication wouldn't control them. And again, this is a brief little picture of this. And it got so disruptive to his life that finally they decided they were going to remove a couple sections of his brain, one on either side in each hemisphere, and try to stop that carrying of these electrical impulses that were just too uncontrolled and leading to seizure activity. And when they did, the surgery was successful in the sense that his seizures were remarkably reduced. And he just did, he was able to function much more like a typical human, typical person, typical individual who can go through life. However, he was unable to form new memories. And that, of course, was extremely disruptive to him and his life. But he proved to be a wonderful way for doctors and neuropsychologists to begin to really explore where in your brain things are happening in terms of memory, learning new memories, forming these new memories that you can actually consolidate, put somewhere, and call on later on. In your book, you should find a diagram of neural networks, a very simple one. Normally, and this is where I again get frustrated that I'm not in the classroom, I go up to the board and I go through this and I draw out the diagram. I know I have a white board that I could click back and forth from. I find that sometimes more frustrating than it's worth, time consuming, and this diagram is beautifully depicted in your book. So please take it from there. But Sapolsky illustrated this in his book in a beautiful way in terms of that there's layers of neurons, and that once you start putting another layer on, they take information and end up with knowing more. Please do take a look at your book. In fact, I probably could get mine, hold it up. I'd rather do that, but let's just go through. I'm going to put all of these up for now and just have you take a look at that. So let's say we have our first layer, and this is so simplistic compared to what we really have. And each one of those areas, Sapolsky used art, knowing something, some kind of art, Gauguin, one of the big classics. And they get information only from one source. But then the next layer gets information from each of those three. So they build a bigger network. And then finally, more neurons up higher get information from all of these layers, different neurons. And so you have an accumulation of information that's only possible because you have access to what neurons know, and you can integrate that and get a bigger and more complete picture. And as Sapolsky puts in the book, most neurons in your cortex have knowledge from the convergence of many, many sources of information. And this is a really important concept. If you disrupt that communication at a different level, you're never going to have that complete picture. Your cortex, neurons in your cortex are never going to know as much as they would otherwise. I am going to leave it up to you to take a look at your book for that. I think it'll be much more efficient. And move on to the neurochemistry of memory and the major neurotransmitter. Glutamate. He's so excited. He's so excited. He's running around. He gets hyper excited by it. He's so excited. Glutamate. Remember that. It's the major neurotransmitter. It's excitatory associated with memory. It's critical for memory. And the synapses are nonlinear in function, meaning you don't just have like, okay, here is one neuron and then it goes to this neuron and this neuron, and it's just a straightforward, there's a combination. There's an effect going to other neurons, much like what you see in this idea of the diagram of the Hubel and Weissel, who were two very, very prominent scientists and gave us so much of this information that we would not otherwise have. This next point is one of the most important for you to remember from this lecture: long-term potentiation, or LTP, depends on glutamate and glutamate receptors. Okay, first, you have these two different kinds, and you have to pass a threshold of glutamate concentration before the second neuron will get excited. Woo-hoo-hoo, I'm firing, and I'm going to help remember something. And when there has been enough of these super excitatory, the synapse gets more excitable, okay, and requires less of an excitatory signal to get an aha. It's like, and first, think about the aha moment. You can hear something, your professor's trying to describe something and says it one way, and you're sitting there, you're like, oh, I'm not getting this at all. Oh man, try to think about it. And then you take that information, and then maybe it's an entirely different professor says something about it in a slightly different way. And you're like, yeah, I think, yeah, now I remember A over here from Professor A. Now Professor B saying this, and I'm adding that on, and it's like, starting to make sense. And now Professor C comes along, says something, and it's like, oh, I get it. And you have an aha moment. Well, with these neurons, you need to get the stimulation, and they get excited, and they get a little more excited, and all of a sudden it's like, woohoo, I'm firing. I've got it. I fired. And I'm going to provide a couple of resources for you to take a look at these receptors that I hope will help you understand a little bit more about them. But just remember that you have to have enough. It's not like a little glutamate. Cooper, come here. He heard me getting excited and he thinks somebody might be here. And of course, it's his job to protect us all. Anyway, after you have this aha moment and you have a synapse, neuron 1, neuron 2, okay? And you're trying to get it. So you only have to stimulate this neuron, release a little bit to get this neuron to go, woohoo, woohoo. And that's your strengthening. So at first, it's like, come on, come on, come on, come on, get going. Not exactly, but you know, you get the idea. Get going, get going. And finally goes, okay, I'll fire. And then this is all via these 2 kinds of glutamate receptors, by the way. And then finally it's like, okay, come on, come on. Okay. And then all of a sudden there's this strength in the synapse. It's like now all this neuron has to do is go, and it's like, I'm firing. So it takes less stimulation, less excitation, less provoking, poking at that memory, or forming a memory. Okay. And the strengthening of a synapse is the potentiation, the P part of LTP, long-term potentiation. I know many of you have had this in other psych courses. I hope I don't go over too quickly for you. But I also don't want to belabor it too much because I know, I would say the majority of you have a sense of what long-term potentiation is. So I do also have this little link here for you that shows a scientist who discovered a lot by using rats what, let me see if I can get here, what happens during the stress, whoopsie, open link, during stress, what can happen to the memory. And it's just a little short, brief link video. And let's just take a quick look at it. I hope. Come on, you. Toilet cleaning. Okay, that was a little clip, hopefully was useful in describing that. And actually, back in my college undergraduate days, I used that swimming test called the Morris test, but with rats. Oops. We sure want that to Hold on. Ah, excuse me, and I will get this off. And hopefully end this. This is one of the reasons I have decided it's not always worth doing these. And as you can see, um, that's why sometimes it's just better just to have you do it. Sorry. Anyway, I did the Morris test with rats, and it is true. They can swim for a very long time, like 30 hours even, but not that they would want to. And if you put a glass, like little pedestal underneath the water, so it's just under the top of the water, they can't really see it, but once they get there, they can get up on it and it gets them out of the water. And they can learn very quickly by looking around and seeing what's in the room and associating a certain position in the pool with that point. And so you can put them in, they'll swim, you can put them in a different place, they'll use the navigational cues and find that and get out. And so that's kind of what this particular researcher was talking about. So let us now just go back here and go back to talking about memory during stress. Now, go back to thinking about sympathetic arousal. Remember that the sympathetic nervous system, before the hypothalamus even starts saying, come on, let's get ready to have the adrenal glands kick out some cortisol, is releasing epinephrine and norepinephrine into the blood. If you actually give a drug called propranolol, which is a beta blocker, some of you may know somebody who takes some form of beta blocker for a heart issue. It blocks sympathetic activation, and you can actually block any effect of the stress on memory. And remember too, again, we have that short-term improvement in memory and then we have the diminished memory. So what happens again? Remember, sympathetic arousal, increase glucose in the circulation. And this helps to fuel the brain activity. The brain is really only like 3 to 5% of the body weight, you know, depending, but it uses about a fifth of body energy. Glucose metabolism is critical, and your brain, your body will work so hard to keep providing your brain with glucose when everything else might be getting deprived, just to keep your brain healthy, alive, and functioning. So a mild elevation, again, going back to that short-term effect, a mild elevation in glucocorticoid levels actually helps memory. They have kind of a local effect on the hippocampus. So now think about this, that if they have an effect on the hippocampus, that means the hippocampus has receptors for glucocorticoids. And this can actually help increase the potentiation. That's why sometimes things are cemented more in your memory than they would be other times. However, prolonged stress, not good. Does not make the hippocampus happy at all. High levels of glucocorticoids actually begin to disrupt that potentiation, that strengthening of synapses. That's so critical for taking memories and placing them in the cortex so you can access them later on. That sympathetic arousal of the norepinephrine and epinephrine, those and other aspects of sympathetic stimulation also begin to disrupt the potentiation. In fact, it can get to the point where the opposite is happening. This prolonged stress with the heightened glucocorticoid levels and norepinephrine and epinephrine and other hormones that are being released under these circumstances actually enhance or lead to something called long-term depression. More ready forgetting. That's something none of us want. We actually end up with the inhibition of glucose delivery. there have been studies and that have shown that hippocampal neurons are actually taking up like 25% less glucose under situations of extreme stress, and then this neural endangerment. You don't want to lose the ability to tap into the long-term memory, what you've already put in your cortex. You want to be able to retrieve this information. And again, remember that for a short time after the stimulation, you actually have heightened memory and recall. You can remember how to get back to your cabin on these windy, crazy paths to get away from the bear and get safe. So as Sapolsky says, get anxious about 15 seconds before your final exam begins and not too far ahead. Good luck if you can do that. You can have a class and help us all do just that, get anxious at exactly the right time. So I mentioned a couple of minutes ago about neuronal damage. Neuronal processes, those axons and the dendrites, think about the cell body and the branch-like processes that emanate from that. They need to make connections with other neurons. So neuron 1, axon, terminal buttons, neuron 2, dendrites, cell body. We want neurotransmitters being released from neuron 1 here to cross the synapse and interact with receptors on the postsynaptic neuron, right? Okay. So we want them in close proximity. If we want these neurons talking, they need to be in close proximity. Under extreme or chronic stress, these can actually shrink. They shrink up, and which if they get smaller, they're going to separate more. So the connections get pulled apart. It's been shown that in rats and humans, that these can regrow at the end of the stressor. However, there are situations in which they don't. If you have a massive stroke, for example, that affects the health of specific neuronal populations in your brain, they may never be able to grow back. Alzheimer's, as unfortunately many of you probably know too well, really tends to lead to a permanent loss of memory. And during this process, you need to tap into more and more associative cues. You've all done that even at this level, and none of us have the situation where we need to worry about never being able to remember anything again. But it's like when you're trying to think of an actor's name, and you go, oh, he was in, what movie was it? Oh yeah. And then you keep going back. Who else was in them? You start getting more and more associative cues, or you could be going back the other way. But that's the thing, those other things that can trigger a memory or help you go through those layers and get the detail you want. Cushing's syndrome. Cushing's syndrome is a situation where you actually have too much of a release of glucocorticoids over a prolonged period of time, and that can actually lead to memory deficits. And I'm sorry, that's probably not on that page in the most recent book. I neglected to go back and correct a few of those pages, but you can find it in the memory chapter. Anyway, glucocorticoids that are being released way too consistently with this particular syndrome, in the absence of any undue stress, in fact, can lead to these memory deficits, and that is called Cushing's dementia. Glucocorticoids over time can lead to the inhibition of the ability of neurons to store glucose. Remember, we want to be able to store glucose so later when we need it, we can utilize it. And remember, that's one of the reasons that under stress, you stop storing because you want to take advantage of what you have at that time. So if you use up all your stored glucose and you can't store anymore, you're going to cause neuro endangerment. Your neurons basically get weaker, like just not so sturdy, not so strong. So if there's another insult, which could be anything like an epileptic seizure, cardiac arrest, a stroke, even concussions. And we're learning all the time how serious concussions, even small ones, especially when they accumulate, they can be very damaging. So if you have these neurons and they're just trying so hard to do their job and they're hanging out and they're just being assaulted by glucocorticoids, they're like, please stop, stop. And they get weaker and just, not sturdy. And then something comes along and deprives them of oxygen and glucose, or perhaps overstimulates them, as it happens in an epileptic seizure. They can actually die. And the glucocorticoids being released all the time impair the survival after exposure to things like the beta amyloid protein that's plays a big role in the development of Alzheimer's, or in the GP120, which contributes to the development of AIDS. So this level of glucocorticoids in your brain is so important to understand the implications. And it's actually been found that the higher the glucocorticoids are when a person enters the emergency room, it appears that there's likely to be more damage and memory problems later on in many of the patients that they've investigated. And we all know that very often people take glucocorticoids for some kind of therapy. Dexamethasone, you're probably hearing about that now in relationship to COVID. It's a treatment to try to help the inflammation. But that's really glucocorticoids. It's our bodies going, oh, this is cortisol, similar. And they are actually used for different disease processes. They block edema due to tumors and other kinds of trauma. They're used in treating flare-ups of MS and other forms of disorders. But if this goes on or frequent, it's possible that they could eventually cause more neuronal damage. And because of that, they should not be used unless absolutely necessary in stroke patients. I think if you put together those facts of what we've been talking about here, you can understand why that would be true. So GCs, I just put there, stands for glucocorticoids, given, it's supposed to be given to AIDS patients, may actually endanger the hippocampus. Furthermore, we've seen where if you take an adrenal gland out of a rat, adrenalectomize them, or you could give them a drug to shut down their own adrenal cortex, you actually can have less damage to the hippocampus after a stroke or a seizure. So what we're basically saying at this point is that you can get weakened, the glucocorticoid levels, the cortisol. Come here, buddy. Cooper says he has high cortisol levels right now because I've been sitting here too long, right, buddy? Anyway, that you can get this weakened, kind of sickly neurons, and if you have truly prolonged exposure to a stressor, you end up with glucocorticoid neurotoxicity. Come here. See if you can get in this picture. And there's Cooper. Hi. Cooper says, don't be worried. Don't be worried. Thank you. I'm all better. Thank you. Okay. Anyway, sorry. So glucocorticoid neurotoxicity. That means that glucocorticoids can actually kill hippocampal neurons. We don't want that. All of this means we have some important questions to answer. Okay, when do we really want to use glucocorticoids? And is there something we can do to buffer their effect on our brain and other tissues, obviously, in our body? But we want to take care of our neurons. We want them to be responsive to short-term lower levels of glucocorticoids, but we need to be aware of the fact that over the long term, excessive glucocorticoids do have the ability to impair neuronal function, the transfer of memories, long-term potentiation, and so forth. Now, one of the negative side effects, let me put it that way, of this course is that you can start thinking about all these things and go, oh my gosh, I'm a mess. Oh my gosh, I bet I'm not going to remember like this or that or the other thing. Oh no. But I know you can sit there and start thinking about all the things that could be happening in your body as a result of stress or long-term stress. Know that just because these things can happen over the long term and excessive stress or chronic stress, that most of us are going to be able to modulate that. And being aware of it, I think, is just so important. It empowers us. The more we know about these processes, the more we should feel empowered to take control and to be able to make some choices that will help us remain healthier longer.